Our next speaker, Mike MacArthur from Zurich, who will tell us something about a new wonder drug, maybe, I hope.
Thank you, Morten. Yeah, like Morten said, I'm Mike. I'm from ETH Zurich in the lab of Jay Mitchell, and today I'm going to tell you a story about a novel compound that we've been using based on the natural compound fumagilin and studying it in the context of weight loss. So this project started out actually as a therapeutic for Prader-Willi syndrome, and PWS is a form of genetic obesity. The genetic etiology of the disease is actually pretty well characterized. It's due to paternal silencing in a specific locus of chromosome 15. But because the locus is relatively large and there's not any obvious kind of coding candidates in that region, there's not really been any traction made in gene therapy efforts. It's characterized by extreme and constant hunger and insatiable appetite, and this manifests in morbid obesity. So this individual with Prader-Willi syndrome is only 5 years old, and this morbid obesity is persistent throughout life. Individuals with PWS rarely live past the age of 30 years, and common causes of death are due to heart failure, respiratory failure due to crushing, and actually overeating to the point of intestinal rupture. So the prognosis for this disease is very poor. There's been no progress made in the therapeutic treatment of the disease, despite many dozens of candidates going through the clinic and all ultimately failing. This was until around 2015 when a company in Boston called Zafgen brought their compound Beloranib into the clinic for PWS. This is results from a phase 2 trial in individuals with PWS, and what you can see is that in the high dose, 2.4 mg of Beloranib, there's about a 10% delta in body weight compared to the placebo. This is a huge result. It was the first pharmaceutical that had ever caused clinically meaningful weight loss in PWS. So there was loads of excitement with this compound. Unfortunately, there was adverse effects related to clotting, and there were 2 deaths in the trial due to pulmonary embolism. So this compound was ultimately clinical development halted, but this is where we came in. Because the results were so profound, we ended up collaborating with Zafgen to understand better the molecular mechanism of action of the drug to try and develop safer alternatives that could harness the same mechanism. So a little bit about the putative target of the inhibitor. The target is MetAP2, so the whole class of fumagilin derivatives are MetAP2 inhibitors, and this protein sits on eIF2 alpha on the ternary complex, and its function is to cleave N-terminal methionine off of nascent peptides as they're being translated. So our goal in this project was to basically characterize second-generation fumagilin derivatives for obesity and to understand what the physiologic and molecular mechanisms of action are. So to start off, we took this new second-generation compound, which we call 1062, and treated obese mice using a subcutaneous dose for 1 month. And what we saw was that there was outstanding weight loss. There was about 25% reduction in body weight, and it was sustained. So as Sarah showed yesterday, we see that it's sustained beyond a month, and actually even into 2 years of treatment, we have a sustained reduction in body weight. This is primarily driven by reduction in fat mass, as we can see a significant increase in the lean to fat ratio of the treated animals. And along with this reduction in body weight, we see reversal of all of the typical phenotypes of obesity. So a reduction in hepatic steatosis and lipid loading, and an improvement in insulin tolerance. But this is probably all secondary to weight loss, so the real question is, what is the mechanism of weight loss? When we think about this in a really simplified model, we can say that a change in fat mass is going to be a function of the difference in energy intake and expenditure. So when we look at energy in these animals, we see that there is no increase. And in fact, over many experiments, we see that there actually tends to be a decrease. But when we look at the change in energy intake, we see consistently an approximately 10 to 15% reduction in food intake over the course of the study. When we look at the kinetics of this change in food intake a little bit more, so here's a shorter term study where we dose for 10 days in the orange box and then cease dosing. We see that the body weight drastically reduces about 20% in just 10 days. And when we take away the drug, when we stop dosing, we see a rapid rebound in body weight. And this corresponds very nicely with a rapid decrease in food intake over the course of the study period, followed by a very rapid and actually compensatory rebound, fueling the regain in body weight. We were able to focus in on this even a little bit more in terms of the temporal scale of reduced food intake in this fasting and refeeding model where we take obese mice and we fast them during the light cycle. So for about 12 hours when they're normally not really eating anyways, we take the food away. 1 hour before we refeed them, we give them a single dose of 1062, we refeed them and we measure their food intake. And what we can see is that in both male and female mice, when we dose before refeeding, that 1 hour is sufficient to reduce food intake again by about 10 to 15%. So our conclusion here is that we have a very rapid reduction in food intake that's driving most of the physiologic benefits we see. And interestingly, when Sarah was able to cannulate these mice in the lateral ventricle, and we repeated the study design, but delivering drug directly into the brain in the lateral ventricle, we were able to reproduce these results. So we think that probably the relevant target tissue is the brain. Next, we turn to some molecular techniques to understand better what the kind of underlying molecular changes driving this reduced food intake may be. So here we just have a RNA-seq experiment where we looked over a time course of treatment in HT1080 cells. And we were particularly interested in these early time points, 4 and 8 hours, because we know that in animals, that's the relevant time frame for the effect. What you can appreciate from these volcano plots is that there's actually not that much changing in those early time points. So we thought that's kind of interesting, and we focused in on these genes which are changing, performing a clustering analysis, and we can see 3 clear patterns. Pattern 1, which is a sustained increase in expression, 2, which is a transient increase, and 3, which is a sustained reduction. And these corresponded very clearly to a single biological process, and this is p53 signaling and cell cycle. So basically this drug is strongly activating the p53 signaling pathway and reducing cell cycle, and it's not really having much else in terms of transcriptional signature. So in following up on this, there's a few dots in between, but to skip to the interesting part, we took p53 knockout animals, whole body knockouts, and tested whether we still have the effect of the drug on body weight and food intake in these knockout animals. And what you can see here is that where the wild type animals have the typical reduction in body weight over the study course, the p53 knockout animals do not. So over this 9-day experiment, the p53 knockout animals do not lose body weight. And this corresponds to a lack of reduction in food intake in the knockouts as compared to the wild types. So this told us that p53 is required for the reduction in food intake and subsequent physiologic response of the drug. Now, what is downstream of p53? Why would p53 be key for this, and what might it be doing? We came up with a pretty good target downstream of p53 when we went back to the transcriptomics. One of the top upregulated genes at both early time points was GDF15. And we found this quite interesting because a lot of recent work has shown that GDF15, which signals exclusively through its brainstem receptor, GFRAL, causes reduced food intake. It's an aversive peptide. And GDF15 is key for the food aversive responses to metformin, chemotherapeutics, and essential amino acid deficient diets. So we think this is quite an attractive candidate for what might be downstream of p53 in our drug response. We went to some cell culture models to confirm this. Here in wild type and p53 knockout HT1080s, we see that the induction in the GDF15 transcript is p53 dependent. And if we go back to the mouse study, we can see that in the serum of these animals, in the wild types, in both a dose and time-dependent manner, we see an increase in GDF15 in serum in response to the drug, whereas we don't see such an increase in the p53 knockout animals. So this is our key downstream candidate in our model, that p53 activation by the drug induces GDF15 to cause reduction in food intake. Our next key question was, what's upstream of p53? Why is this drug activating p53, and what could be kind of the molecular wiring that's facilitating the induction. We did a lot of testing to determine whether the drug might be genotoxic, because that's the classic p53 activating stimulus. We don't see evidence for genotoxicity really at all in our cell or animal models, especially at the relevant doses. So to kind of get at this question, we performed a genome-wide positive selection CRISPR screen, and this is really a prime candidate for this technique because our readout is reduction in proliferation. So essentially, our drug reduces proliferation of cells. When we infect cells with our genome-wide CRISPR library and then treat with the drug, we expect that cells resistant to the anti-proliferative effect will be our genes of interest for p53 activation. So we used this genome-wide library. We performed the experiment in 2 cell lines, and we performed it at 2 time points, 7 and 14 days. So basically 4 independent experiments. And we saw some really nice results here. So what you can appreciate from the 4 independent experiments is that the top hit across all is p53. And this tells us the experiment works because we know that the cytostatic effect of the drug has to go through p53 from previous experiments. We also see some really interesting candidates. So in all 4 experiments, we see the same 2 candidates popping up, also with very high degree of statistical significance in their guide enrichments and showing guide enrichment across all 4 unique guides corresponding to the genes. So when we run the statistics across the 4 experiments, we see that 3 genes pop up as being significantly enriched in the resistant cells. They're p53, and they're 2 targets related to Hippo signaling pathway. So this was quite interesting to us. This was a pathway that certainly wasn't on our radar as being important in the response to the drug, but the screen makes it quite clear that probably activation of Hippo signaling pathway is driving the p53 activation. And we actually went back to some older data where we've treated mice with the drug at a very young age, and what we can see is that the mice who are treated with the drug from 5 weeks of age and let grow into adulthood actually have a much smaller body size. And this is consistent with the classic action of Hippo signaling pathway, which is regulation of body size. When it was first discovered in Drosophila, activation of Hippo signaling, as here, causes a small body size in the flies. And we quantify this by body length and tibia length. We see that the reduction is quite profound. So kind of our final question now is, what does MetAP2 have to do with any of this? How does an inhibitor of this enzyme that's responsible for N-terminal methionine aminopeptidase activity interact with Hippo signaling pathway and activate p53? We're not exactly sure. We've developed some tools. So we have knockout cell lines that we can use to address this question. And MetAP2 is a really difficult target to work with because it's essential in vivo. It's also conditionally essential. So even knocking it out in endothelial cells and in neurons causes in utero death in mice. So it's really difficult to study in vivo, but we've generated some tools in vivo to knock it out in the brains of adult mice. So this is kind of helping us discover what the role of MetAP2 might be here. And then finally, this is a derivative of a natural product. And natural products often have more than one target. So we're working in collaboration with a group at Harvard Medical School to determine if there are additional targets besides MetAP2 that may be driving some of this response. So this is a thermal proteome profiling experiment where we do a proteome-wide assessment of binding by the compound. We can see here in the circle is MetAP2, so it's definitely the most robust binding partner, but we also see other hits which look quite promising and may be giving us a non-MetAP2 dependent effect for the drug. So in summary, the 2nd generation fumagilin derivative we've been working with, 1062, reverses obesity by reducing food intake in a robust and rapid manner. That reduction in food intake is driven by p53 activation and probably downstream by GDF15 induction. The Hippo signaling pathway plays a critical role in the activation of p53, and the exact role of the MetAP2 as the target of the drug is unclear but under investigation. So I'd like to thank our lab. I'd like to thank especially Sarah Mitchell, who's been key in a lot of these experiments, our industry collaborators for the financial and compound support, and then I'd like to thank Morton for having us and organizing this great conference. Thank you.
Thank you, Mike. Maybe one quick question. I think it's fascinating that you have a DNA damage response enzyme that regulates food intake. Do you think that we're able to taste stuff that damages our DNA? Or how is thinking about this in the physiological context?
Yeah, tasting is unclear, but for example, the essential amino acid deficient diets, if you take leucine out of the diet, you can't really taste it, but for some reason there's definitely activation of signaling that causes you to eat less of it. So I think in a way you can subconsciously taste it, yeah.
It's very fascinating. I think we have to move on, unfortunately.